Hi my dear students, welcome to my biology class. In the previous class, we have learned about the structure of a polynucleotide chain. Today we are going to learn about another topic, that is DNA. You might have seen this figure. This is the double helical model of DNA. And what is DNA? DNA is the genetic material in most of the living organism except in few living organisms. DNA is a polynucleotide and it is also a nucleic acid. So today we are going to learn about the salient features of DNA. So what is the expanded form of DNA? Deoxyribo nucleic acid. It is a polynucleotide. The DNA is actually the chemical nature of the gene. Genes are the unit of inheritance which transmit the characteristic features from the parents to offspring. So, DNA has functional segment as well as non-functional segment. The functional segments of DNA are the genes. So, we can say the chemical nature of the gene is the DNA. Before I begin with the salient features of the DNA, I have to share with you the historical background of DNA. DNA as an acidic substance present in the nucleus of the pus cells was first isolated and identified by Frederick Miescher in 1869. Frederick Miescher. In 1869, he isolated the acidic substance from the nucleus of the pus cells. Okay, then he called it as nuclein. He named it as nuclein. Later, the acidic substance was renamed as nucleic acid by Altman in 1889. Later in 1953, James Watson and Francis Crick proposed the double helical model of DNA. This model has two strands. So it is arranged in a specific fashion, a right-handed helical fashion. This double helical model of the DNA was proposed by James Watson and Francis Crick. Double helical model of DNA. How they proposed the double helical model of DNA? That was based on the X-ray diffractions made by Maurice Wilkins and Rosalind Franklin. The two scientists, they made the X-ray image of the DNA. So with the help of the information, they proposed, James Watson and Francis Crick proposed the double helical model of DNA. Then who made the X-ray diffractions? X-ray diffractions was made by Maurice Wilkins and Rosalind Franklin. Now let's study the salient features of DNA which was proposed by James Watson and Francis Crick. They proposed a double helical model of DNA. Double helical model. Here double helical means this DNA has two strands. That means two polynucleotide chains. Two polynucleotide chains which are arranged in a specific manner. Imagine a spiral stair, how it is oriented or arranged in a right-handed fashion, how it is twisted in a right-handed manner. Similarly, the two strands of DNA are arranged in a specific manner in a right-handed fashion. So it is a right-handed helix. Okay, so polynucleotide chains means as it is a double-stranded structure, it must have two polynucleotide chains. We have already learned the structure of a polynucleotide chains in the previous class. In the polynucleotide chain, there are many nucleotides. A nucleotide is composed of deoxyribose sugar, nitrogen base and a phosphate group or phosphate moiety. Now let us recall the structure of the polynucleotide chain, the structure of DNA. Then suppose this is the pentose sugar with a 5 carbon atom. First, second, third and fourth positions of carbon atom. Fifth position is here CH2, and it makes a link between the phosphate group and it is represented as P double bond O OH. This is the free phosphate that is attached at the fifth carbon atom of the pentose sugar. Then at the first carbon atom, the sugar is connected with by N-glycosidic link to the nitrogen base. Suppose one nitrogen base adenine. Okay, adenine. So this is the typical structure of a nucleotide in the DNA polynucleotide chain. Ribose sugar, phosphate group and one nitrogen base. How this nucleotide is connected to the next by means of phosphodiester bond. Here from the fifth carbon atom, it forms as a bond. It makes a bond between the phosphate group by means of phosphoester linkage, but one ribose sugar, deoxyribose sugar is made a bond between the next nucleotide by means of phosphodiester linkage. Now let us understand how is it possible. Next, deoxyribose sugar, its fifth position CH2, and this is the first position, and suppose let us imagine guanine is connected or attached with this. Okay, then how it forms as a bond in between the 3' position of the carbon atom and the fifth position. In between there is a bond, that is phosphodiester bond. Okay, phosphodiester bond. This is called a phosphodiester bond. This is one side of the polynucleotide chain. In this case, the fifth position is free with the phosphate group. So this strand begins with the 5' polarity. And at the end, it has a free OH group, hydroxyl group. So it is called 3' end. Okay, this is one side of the double-stranded DNA. The second strand begins with a 3' and let's see how is it formed. Then let me draw one deoxyribose sugar. Here we have to draw the sugar in an upside down fashion. Okay, first position, second, third, fourth, then fifth. Here the first position has a nitrogen base, that is thymine. Okay, then third position OH is free, it is 3' end. Then this is connected to the next deoxyribose sugar, next nucleotide by means of the same bond, that is phosphodiester bond. Let me draw the second one also. First, second, third, fourth, fifth. Okay, here fifth carbon atom has a connection with the third carbon atom of the next nucleotide. So it forms as a phosphodiester link. Okay, phosphodiester, P double bond O here, O here, O here. Okay, pentavalent phosphate group. Here the phosphodiester linkage. So here also at the first position there is another nitrogen base, that is cytosine. Got it? So in this figure you can understand that the first strand has the polarity like this, 5' to 3'. In the second strand it begins with a 3' end, and look at here, it has a free phosphate group, so that it can be shown like this. Okay, free phosphate group, so that it is 5' a free phosphate group at the fifth carbon atom of the nucleotide. So what can we understand from this figure? Here the two strands are arranged in an anti-parallel orientation. So the second point, two strands are in anti-parallel orientation with the opposite polarity. That means if one strand has a 5' to 3' polarity, the other strand will have a 3' to 5' polarity. That is the anti-parallel orientation of the two strands. Here the sugar and phosphate act as the backbone of the DNA. Look at here, this is the deoxyribose sugar, the first one, second one. Here the phosphate group forms the link between the two nucleotides. So let's say that the sugar and phosphate groups form as the backbone of the DNA double strand. Sugar and phosphate form as what? Backbone of DNA. Okay, here the nitrogen bases are faced inwards. They project inwards. Nitrogen bases project inwards. Okay, here there is a specific nature in connection with the nitrogen bases of the DNA double strand. James Watson and Francis Crick also remarked on the base pairing of the nitrogen bases in the DNA double strand. According to base pairing rule, adenine always pairs with thymine by means of two hydrogen bonds. It can be represented like this: adenine pairs with thymine by two hydrogen bonds. Because it is easy to make a hydrogen bond with the adenine and thymine. That's why it forms as a bond in between these two nitrogen bases. So it can be represented like this. Okay. Then here the hydrogen bonds are very weak, but it gives the structural stability to the DNA double stranded structure. Similarly, guanine is another nitrogen base. It always pairs with the cytosine by means of three hydrogen bond. Here the guanine always pairs with the cytosine by three hydrogen bond. So adenine pairs with the thymine by two hydrogen bond. Guanine pairs with the cytosine by 3 hydrogen bond. Vice versa, thymine to adenine by 2 hydrogen bond, cytosine to guanine by 3 hydrogen bond. So here there is a specific nature in connection with the nitrogen bases. Look at the figure. Suppose this is adenine and this one is thymine. And the next one, guanine, cytosine. Here one nitrogen base talks over the other. Suppose this is one nitrogen base, that is adenine. It talks over the thymine. Okay, so the arrangement of the nitrogen bases like this inside the two strands of the DNA provides additional structural stability to the DNA, and also it maintains an equal distance between the two strands. How is it possible? We know that adenine and guanine are purines, double ringed structure, while cytosine and thymine are single ringed structure. Purines always pair with the pyrimidines. That means when a double ringed structure is present in the DNA, the opposite nitrogen base will be a single ringed. So that in the next pair, look at here, adenine is double ringed, thymine is single ringed. Here guanine is double ringed, cytosine is single ringed. So that the distance between two strands can be maintained as approximately equal in between the two strands. As adenine pairs with the thymine, guanine pairs with the cytosine, this is called base pairing. Base pairs. This phenomenon is called base pairing. These are called base pairs. Adenine thymine is one base pair. Guanine cytosine is another base pair. So similarly, there are many base pairs in the DNA double helical structure. Next, let's understand the pitch of the DNA. The full turn of DNA is considered as its pitch. It is about 3.4 nanometer. 1 nanometer is equal to 10 raised to minus 9 meter. 1 nanometer is equal to 10 raised to minus 9 meter. That is the 1 billionth of a meter. 1 nanometer is equal to 10 raised to minus 9 meter. Then here in the full turn, there are approximately 10 base pairs. So each step can be considered as one base pair, like guanine, cytosine, adenine, thymine, that cytosine, guanine, like that it forms as base pairs. In the full turn, there are approximately 10 base pairs, so that the distance between two consecutive base pairs is 0.34 nanometer. Two base pairs is equal to 0.34 nanometer. How can we find out the distance between two consecutive base pairs? It can be found out by dividing the pitch by the total number of base pairs. Here the pitch is 3.4 nanometers divided by 10 base pairs in full turn. So that the answer is 0.34 nanometer. The distance between two consecutive base pairs is 0.34 nanometer. Look at the figure. This is the double-stranded polynucleotide chain. It is actually the diagrammatic representation or the cartoon figure of the polynucleotide chain, that is DNA. DNA has two strands. The first strand is with a 5' to 3' polarity. The opposite strand has 3' to 5' polarity. Look at here in the first strand, the 5' moiety, the phosphate moiety is free, so that it is called 5' end. At the end, there is a free OH hydroxyl group, that is 3' end of the first strand. The next strand is called complementary strand. Complementary. Complementary. These two strands are complementary to each other. That means if we get any strand, that means the nitrogen bases of any strand like this, adenine, thymine, guanine, we can easily write its that complementary base based on the base pairing rules, such as adenine to thymine, thymine to adenine, guanine to cytosine, so that both strands are complementary to each other. Okay, so look at the second strand. The strand begins with the 3' and ends with the 5' end. 3 dash position there is a free OH group, at 5 dash position there is a free phosphate group. The complementary base pairing of the double helical strand is also based on the observation made by Erwin Chargaff. The rules that are based on the complementarity of the two strands was proposed by Erwin Chargaff. This rule is called Erwin Chargaff's rule. According to him, the two strands of DNA has complementary base pairs. Complementary adenine thymine, thymine adenine, guanine cytosine, cytosine guanine, like this complementary base pairs. According to him, the percentage of or the amount of adenine molecule is equal to the amount of thymine molecule, because adenine is a purine, it always pairs with the thymine, the pyrimidine. Similarly, the amount of guanine is equal to the amount of cytosine. Here it means that suppose if there is 30% adenine in a particular DNA molecule, the same percentage that the thymine will be there in the same compound, same DNA molecule. So what would be the percentage of guanine and cytosine? Is 30 plus 30 total 60. The remaining 40% is shared by guanine and cytosine equally, so that 20% guanine and 20% cytosine. So this is the way the amount of that adenine is equal to thymine, amount of guanine is equal to cytosine. So the total percentage will be 100. 30 plus 30 plus 20 plus 20 is equal to 100. Or it can be written like this: adenine plus guanine is equal to thymine plus cytosine, or adenine plus guanine by thymine plus cytosine is equal to 1, or a constant. Here it is clearly you can understand that if adenine has 30%, guanine 20%, thymine 30%, cytosine 20%, 50 by 50, that is equal to 1. This is known as Erwin Chargaff's rule. The amount of adenine is equal to thymine, the amount of guanine is equal to cytosine. This particular rule is applicable only to the double-stranded DNA. There are single-stranded DNA, but it cannot be applied in single-stranded DNA. It can be applied only to the DNA with a double strand, the double-stranded DNA that shows base pairing. Okay, dear students, today we have learned about the historical background of the DNA, the salient features of DNA, which was proposed by James Watson and Francis Crick, and also Erwin Chargaff's rule, complementary base pairing rule. I hope you have understood the topic well. With this, I conclude my session. Thank you and have nice moments.